Hello students. In this lecture we are going to discuss genetic point, that is techniques involved in recombinant DNA technology. Among that we are going to discuss blotting techniques. There are different types of blotting techniques like southern blot, northern blot and western blot. Now southern blot, it is used to analyze or detect DNA. Northern blot, it is used to detect RNA. Western blot, it is used to detect protein. So in this lecture we are going to see southern blotting, which is used to detect or analyze DNA. Before going to the southern blotting, let's understand what is blotting. Okay, so blotting, it is the transfer of macromolecules like DNA, RNA or protein from gel onto the membrane. Okay, that is the blot, and that process is known as the blotting process. Then that membrane may be nitrocellulose membrane or nylon membrane. Mostly nylon membranes are used. The reason behind that is there is strong interaction of macromolecules with this membrane. Strong interaction means they bind firmly to that membrane. Okay, so we use mostly nylon membrane. That is the blot, or we can say blotting process, that is only included in this presentation. Blots are techniques for transferring DNA, RNA and proteins onto a carrier so they can be separated easily. The scientist Edward Southern in the year 1975 developed this technique and is widely used to analyze DNA fragments. Okay, so the name is given Southern blotting. There are three basic requirements for this blotting technique. The first is your DNA fragments, which is being separated by agarose gel electrophoresis. Means this technique, southern blotting technique, involves agarose gel electrophoresis technique. Okay, in which the DNA molecules are being separated. So first requirement is AGE, that is agarose gel electrophoresis, DNA fragments separated by AGE. Second, your nylon membrane or nitrocellulose membrane for fixation. Third, probe. Okay, third requirement is your radio labeled probe. What are probe? Probes are short oligonucleotide sequence which are radio labeled. Okay, that are probe. और ये जो probe की sequence होती है, that is complementary to our DNA sequence, or we can say to our gene of interest sequence. So that is probe which has been radio labeled, या फिर वो fluorescently labeled होते या radio labeled होते. Okay, so these are three requirements. DNA fragments separated by agarose gel electrophoresis. Second requirement is your nylon membrane or nitrocellulose membrane, and third requirement is radio labeled or fluorescent labeled probe. The principle behind this technique is as this technique uses agarose gel electrophoresis. In agarose gel electrophoresis, separation of DNA is based on the molecular weight. Means large molecule will move slowly and small molecule will migrate fastly. Okay, so the same principle is involved in southern blotting. This is one thing. Second principle is use of radio labeled probe. As I told, the sequence of probe is complementary to our gene of interest. That complementary sequence will bind. So the probes will bind to our complementary sequence, and the probe which are not complementary to our gene of interest, they will be washed out. Okay, or they will remain unbound. So that is the second principle involved behind the southern blotting. Let's start with the procedure or steps used in the southern blotting. First of all, in the diagram you can see double-stranded DNA is being shown. In this, now this DNA contain our gene of interest sequence. Okay, gene of interest, what is that gene of interest? It is the gene which we want to study or which we want to analyze. Okay, that is हम उसे represent करते हैं as a gene of interest. तो ये डबल स्ट्रैंडेड डीएनए में कहीं तो हमारा जीन ऑफ इंटरेस्ट प्रेजेंट है. ओके, जिसकी सीक्वेंस हमें नोन है, whose sequence is known and whose restriction site is also known. What are restriction sites? Restriction sites are the sites on to which restriction enzyme shows its activity and cleave the DNA into fragments. That are the restriction sites. तो जीन ऑफ इंटरेस्ट की सीक्वेंस हमें नोन है और उसके रेस्ट्रिक्शन साइट्स भी हमें नोन है. ओके, सो वी विल यूज द रेस्ट्रिक्शन एंजाइम टू cleave this double stranded DNA. In second step you can see, or in the second diagram आप देख सकते कि फ्रैगमेंट्स आर बीन प्रोड्यूस्ड. ओके, डीएनए के फ्रैगमेंट्स प्रोड्यूस्ड है, 3 फ्रैगमेंट्स प्रोड्यूस्ड है. तो हमारा जीन ऑफ इंटरेस्ट इन 3 फ्रैगमेंट्स में से कहीं भी प्रेजेंट हो सकता है, आइदर टू द फर्स्ट, सेकंड और थर्ड. वी डोंट नो वेयर इट इज प्रेजेंट. ओके, हमें वही एनालाइज करना है, हमें वही स्टडी करना है, हमें पता नहीं है. ओके, बट उसकी सीक्वेंस पता है. सो अमॉन्ग दिस 3 फ्रैगमेंट्स वी डोंट नो वेयर आवर जीन ऑफ इंटरेस्ट इज बीन प्रेजेंट. So for that we will carry out agarose gel electrophoresis. So the second step is to carry out agarose gel electrophoresis. So कैसे fragments run होंगे? We will load the fragments onto that gel, okay, onto the well, and the fragments will run on the basis of the molecular weight. As larger molecules slowly migrate होंगे और fast molecule will migrate. Small molecules will migrate fastly and large molecules will migrate slowly. So this is the second step, that is to run the agarose gel or to carry out the agarose gel electrophoresis. In the third step, the separated DNA fragments along with the gel is put up into mild alkali solution. माइल्ड अल्कली सॉल्यूशन कौन सा यूज किया जाता है? NaOH, सोडियम हाइड्रोक्साइड. नाउ व्हाई वी पुट दिस separated DNA fragments along with the gel onto this NaOH solution? Just to convert our double stranded DNA into single stranded DNA. ठीक है, हमें डबल स्ट्रैंडेड डीएनए कन्वर्ट करना है सिंगल स्ट्रैंड ऑफ डीएनए में. इसीलिए हम उस सेपरेटेड फ्रैगमेंट्स को जो जेल में हम सेपरेट हुए, अलॉन्ग विद द जेल वी डीप इनटू दिस NaOH सॉल्यूशन. अब हमें सिंगल स्ट्रैंडेड डीएनए क्यों चाहिए? द रीजन बिहाइंड दिस इज हम आगे के स्टेप्स में हम प्रोब ऐड करने वाले. ओके, सो टू बाइंड टू द प्रोब it is necessary that our DNA must be in the single stranded. Okay, तो इसीलिए हम डबल स्ट्रैंड का कन्वर्जन सिंगल स्ट्रैंड में करते, so that probe can easily bind when its complementary sequence he finds. Okay, so that for that process or for that reason we separate our double stranded DNA into single stranded DNA by dipping this gel along with the separated fragments into NaOH solution. So this is the third step. In the fourth step, we use the nitrocellulose filter or nylon membrane, that is known as the blotting technique. Means what we do, we transfer our single stranded DNA to the nitrocellulose membrane. ठीक है, हम ट्रांसफर करते हैं. So how we transfer this nitrocellulose, or you can say how we transfer this single stranded DNA into the nitrocellulose membrane. So we take the tray. Okay, let me show you in the diagram. So okay, we have transferred our single stranded DNA to the nitrocellulose membrane. How we transfer then to the nitrocellulose membrane, that I'll show you with the diagram. This is the tray. Consider this is the tray. Onto that tray we have put up the buffer solution. ठीक है, उस ट्रे में हमने बफर सॉल्यूशन डाला है. into that tray हमने एक सपोर्ट सिस्टम रखी है. Okay, that support system may be sponge or glass. Onto that support system we have put up our gel. Okay, हमने क्या रखी है? अपनी gel containing single stranded DNA. Okay, onto that gel we have put up our nitrocellulose membrane या फिर nylon membrane. Okay, onto that gel हमने क्या डाला है? nitrocellulose membrane डाला है. Onto that nitrocellulose membrane we have kept Whatman filter paper. ओके, वॉटमैन फिल्टर पेपर रखा है और वॉटमैन फिल्टर पेपर के ऊपर हमने वेट रखा है, कोई हैवी चीज रखी है. व्हाई हैवी चीज? बिकॉज ये एक दूसरे से स्टैक की तरह अटैच रहे, ओके, एकदम फर्मली अटैच रहे, इसीलिए हम कोई भी ऊपर हेवी चीज रख देते या फिर वेट कोई भी हम रख देते हैं, ठीक है। सो हम जो नीचे हमने जो सपोर्ट सिस्टम रखा, इट में बी एनी स्पॉन्ज और ग्लास, ऑन टू दैट वी हैव केप्ट आवर जेल विच कंटेन सिंगल स्ट्रैंडेड डीएनए। उसके ऊपर हमने रखा है हमारा नायलॉन मेम्ब्रेन, उसके ऊपर हमने रखा है वॉटमैन फिल्टर पेपर, और ये सब चीजें एक दूसरे से फर्मली अटैच हो, इसीलिए हमने कोई हैवी चीज ऊपर रख दी है, ठीक है। सो व्हाट विल हैपन आफ्टर सम टाइम, पेपर विल गेट वेट, ओके, पेपर क्या होगा, बफर की एक्शन अपवर्ड होगी, तो उस वजह से पेपर वेट हो जाएगा, वॉटमैन फिल्टर पेपर विल बिकम वेट। वंस द पेपर हैज बिकम वेट, इट इंडिकेट्स दैट आवर single stranded DNA is been transferred into the nylon membrane, ठीक है। एक बार पेपर वेट हो गया तो इसका मतलब ऐसा है कि हमारा सिंगल स्ट्रैंडेड डीएनए इट इज बीन ट्रांसफर्ड ऑन टू दैट nitrocellulose membrane. So afterwards what we do, slowly we take this nitrocellulose membrane separately, okay, we separate that nitrocellulose membrane containing our single stranded DNA, और हम फिर, let me erase this, okay, in the next step what we do, we remove that nitrocellulose membrane, and then for the visualization हम क्या करते हैं, अब वो मेम्ब्रेन हमने उठाया है, नाउ देर इज नो विजुअलाइजेशन, वी कांट सी दैट आवर मेम्ब्रेन, आवर सिंगल स्टैंडर्ड डीएनए इज बीन ट्रांसफर्ड ऑन टू दैट मेम्ब्रेन, हमें वो विजुअलाइज नहीं हो रहे, बट वॉटमैन फिल्टर पेपर वेट हो चुका है, इसीलिए हम समझ चुके हैं कि आवर सिंगल स्टैंडर्ड डीएनए इज बीन ट्रांसफर्ड ऑन टू दैट मेम्ब्रेन, but it's unable to visualize. So for visualization what we do, these DNA bands are transferred, or we can say along with this membrane is transferred to heat treatment और UV treatment, okay, heat treatment दी जाती है या फिर UV treatment दी जाती है, क्यों, because to form that single stranded DNA onto that nitrocellulose membrane, as वो firmly attach हो ही जाते हैं, but to fix, ताकि वो निकले ना, okay, so to firmly fix onto that membrane we provide the heat treatment or UV treatment, okay. After that we add that membrane along with the single stranded DNA to the probe solution, okay, हम probe solution पे वो membrane transfer करते हैं, तो what will happen, अगर complementary sequence होगी, that probe along with the probe क्या है, they are the short oligonucleotide sequence which are radiolabeled, so this probe will get bind to our fragment, single strand fragments, okay, अगर वो complementary होंगे they will bind, if they are not complementary they will remain unbound, okay, so unbound probes are washed out and the probes which are bind they are definitely complementary to our sequence. Then for visualization we perform the ऑटो रेडियोग्राफी, ठीक है, हम क्या परफॉर्म करते हैं, ऑटो रेडियोग्राफी, अगर प्रोब रेडियो लेबल्ड होंगे तो हम ऑटो रेडियोग्राफी परफॉर्म करते हैं, एंड विजुअलाइज आवर जीन ऑफ इंटरेस्ट इज ट्रांसफर्ड और नॉट, ओके, हम विजुअलाइज करते हैं, बैंड्स फॉर्म कैसे, हमें ऑब्जर्वेशन कैसे होता है, बाय द फॉर्मेशन ऑफ द बैंड्स, ओके, आप इस 6 स्टेप में देख सकते हैं, देयर इज, अगर बैंड फॉर्मेशन होता है, देयर इज प्रेजेंस ऑफ band formation, then it indicates that our gene of interest is being transferred properly. If there is no formation of band, then it indicates that gene is absent, our gene of interest is absent. So this is very simple analyzation for the detection or to analyze our gene of interest. If bands are present, it just indicates that our gene of interest is been present or it's been transferred onto that membrane. If bands are not observed, then it indicates that gene of interest is not been transferred, or it shows or it is absent. Okay, so that is the simple procedure for that. डायग्रामेटिकली भी है, यहाँ पर डायग्रामेटिकली भी दिखाया गया है. सी इन द फर्स्ट स्टेप डीएनए इज देयर. वी विल क्विकली रिवाइज कि स्टेप्स क्या है. इन द फर्स्ट स्टेप द डीएनए इज देयर. ऑन टू दैट डीएनए आवर जीन ऑफ इंटरेस्ट इज प्रेजेंट, gene of interest which we want to study or analyze. In the second step, what we have done, वी हैव यूज द रेस्ट्रिक्शन एंजाइम्स. ओके, सो फ्रैगमेंट्स हमें दिखे, उस फ्रैगमेंट्स में हमारा जीन ऑफ इंटरेस्ट कहीं भी प्रेजेंट हो सकता है. वी कैरी आउट एगेरोस जेल इलेक्ट्रोफोरेसिस. बैंड्स आर सेपरेटेड ऑन द बेसिस ऑफ देयर मॉलिक्यूलर वेट, एंड देन that separated bands along with the gel is dipped into the alkali solution like NaOH to convert that double stranded DNA into single stranded DNA. After that we transfer that single stranded DNA onto the नाइट्रोसेलुलस मेम्ब्रेन. तो कैसे ट्रांसफर करते हैं, सपोर्ट सिस्टम, उसके ऊपर हमारा जेल कंटेनिंग सिंगल स्ट्रैंडेड डीएनए, एंड देन ब्लॉटिंग मेम्ब्रेन, उसके बाद वॉटमैन फिल्टर पेपर, एंड देन वेट. ओके, सो वी इन द थर्ड स्टेप वी ट्रांसफर दैट डीएनए. उसके बाद हम क्या करते हैं, वी सेपरेट आउट दैट मेम्ब्रेन, राइट. So for fixation of that single stranded DNA we give the heat treatment or UV treatment. Then we add probes. If they are complementary, probes will get attached, right, radio labeled probes will get attached. And then afterwards for the visualization we use the autoradiography technique. Okay, in autoradiography technique bands are formed. Formation of band indicate that our gene of interest is been transferred or it has been present. If there is no formation of band, then our gene of interest is not present or it is not been transferred. That is the simple technique known as the southern blotting. In detail diagram में which is same as that I told you earlier. If we see the applications for southern blotting, then this is used in the mapping, then developmental study, for the diagnostic purposes, in the forensic study. Okay, secondly it is used to detect presence or absence of gene of interest. Definitely bands से हम देख सकते हैं कि whether our gene of interest is present or not. Okay, or we can say transferred gene is present or not. Southern blot allow investigators to determine the molecular weight of restriction fragment and to measure relative amount of different samples. So these are the different applications which we can use or which we can perform by southern blotting technique for analyzing or to study the DNA fragments. I hope you might have understood this technique, southern blotting. In the next lecture we will see next blotting technique. Thank you.